Hello, my name is Mikhail Gudkov. I'm a PhD student at the Victor Chang Cardiac Research Institute. This work is titled Quantifying Negative Selection on Synonymous Variants. Synonymous variants are defined as single nucleotide variants that replace a codon with a different one, encoding the same amino acid. There are many potential consequences of synonymous variants, but in this project, we're interested in their effect on messenger RNA stability and degradation. Here we used a recently developed metric called MAPS, or the mutability adjusted proportion of singletons, to study the deleteriousness of these mutations. Singletons are variants that are present only in one individual in a population and have allele count of 1. MAPS shows relative intensity of negative selection, using the assumption that if a particular variant is damaging, it will be rare in the population, because purifying selection will be trying to remove it. Higher values indicate an enrichment of singletons, which suggests higher deleteriousness of the corresponding class of variants. MAPS uses mutability correction, which is based on how likely any particular mutation is to occur and on methylation levels to eliminate bias towards CpGs. For example, transversions, that is, mutations from a purine base to a pyrimidine base or vice versa, are more severe events than transitions, and therefore have lower chances to mutate acceptably. Thus, for transversions, we should expect more singletons, because again, such events are less common. We fitted a series of MAPS models on a QC-compliant subset of over 2 million synonymous variants from nomad exome sequences. As you can see in Figure 1, the original model had a tendency to slightly underestimate the real number of singletons for transversions. In order to reduce this bias, we applied a square root transformation, which improved the overall fit of the model. We defined optimality-reducing synonymous variants as mutations creating a codon with a lower codon stability coefficient score than the original one. This score is calculated as the Pearson correlation coefficient between each codon's frequency in an mRNA transcript and its half-life in human embryonic kidney cells. According to this definition of optimality, transcripts enriched with non-optimal codons with a negative codon stability coefficient tend to have higher degradation rates. Using this dichotomy, we determined that optimality-reducing variants are subject to stronger negative selection than optimality-increasing ones, as indicated by the positive difference in their MAPS scores in Figure 2. We then selected variants in 20% most constrained genes using a metric of intolerance called LOF, which is based on the deficit of potential loss-of-function variants in a gene. Our results, shown in Figure 3, provide evidence for higher deleteriousness of both optimality-reducing and optimality-increasing synonymous variants in those genes. Finally, we applied GRP, a metric for conservation analysis between species. Positive GRP scores represent strongly conserved positions, whereas negative scores represent variable positions. For this analysis, we only used 2 codon amino acids, as you can see in Figure 4. Consistent with the hypothesis that optimal codons experience stronger evolutionary conservation, we found that optimality-reducing synonymous variants were shifted towards positive GRP values relative to optimality-increasing variants, which is especially true of glutamine. In other words, optimality-reducing variants correspond to loci with high rates of rejected single nucleotide substitutions. To conclude, our main results can be summarized as follows. Synonymous mutations that reduce codon optimality undergo stronger purifying selection than those that increase it. Negative selection affects synonymous variants in a gene and amino acid dependent manner, with both optimality-reducing and optimality-increasing variants being more deleterious in constrained genes. Among 2 codon amino acids, glutamine has the highest divergence between its optimality-reducing and optimality-increasing classes of synonymous variants based on MAPS and GRP scores. And finally, square root transformation improves the fit of the MAPS model for synonymous mutations. Thank you, and I'm happy to take any questions.